Welcome back to Chapter 13, Part 3. Here's where we're going to talk a lot about DNA technology. And so let's go ahead and dive into some of this really interesting technology. The first one we're going to talk about is how DNA and the very structure of DNA and how it replicates makes genetic engineering possible. So take a look at this strand of DNA right here. This particular strand of DNA is called a probe, and we're going to use this probe, which is usually single-stranded, and it's been built based on a known sequence of DNA. And we're going to use it to hybridize with a complementary sequence. So notice that A, and then all these G's here are going to, if it ever encounters this complementary sequence, it's going to form a duplex with that. And we call this nucleic acid hybridization. So again, nucleic acid hybridization is where you take a probe, a single-stranded probe, and that probe usually has these reporter molecules on it. They're usually something like radioactive probes or something along those lines, some radioactive markers. They could also be fluorescent markers, so it depends. But notice that if you take this probe and you put it with this particular sequence, you will get a perfect hybrid, a perfect duplex. And so if we are trying to identify an unknown organism, we can say, oh yeah, well we know this region of it, so we can identify this region of, we can basically identify it, or we can detect it. On the other hand, if we take the same probe and we put it into a little test tube with this target sequence, notice that they're just not going to match up. And so you don't get a duplex formed. If the duplex isn't formed, then you cannot detect that this bit of DNA is present, because remember, this is this little dot right here and right here, that's our reporter molecules. So they're like, oh, well, I'm not hybridizing with anything, so it's just not detectable. This nucleic acid hybridization really forms the foundation of almost every single technique used in genetic engineering. And we define genetic engineering as the direct manipulation of genes for practical purposes. The first of those practical purposes that we're going to talk about is DNA cloning. So cloning just means to make multiple copies of a gene or some other segment of DNA that's not a gene. I know you think DNA is all made up of genes, and we'll learn the official definition of gene in another chapter. But for now, we could either use a gene or something else, some other region of DNA that's not really a gene. So if you want to work with specific genes, what they're going to do is they're going to work with well-defined segments of DNA. And you need identical copies of that DNA, and so we're going to use cloning. Most methods for cloning involve two separate pieces of DNA. We have the DNA fragment in yellow right here. This is the fragment that we're interested in. And we have the vector. So a vector is not interesting at all to the biologists, except for the fact that the vector can bind to the DNA fragment and make something called recombinant DNA. So those are the bits that we need to have in order to make recombinant DNA. Notice that in the previous slide, our DNA, when the DNA fragment combined with the vector, it made a circular chromosome. And remember that circular chromosomes are found in prokaryotic organisms, like bacteria. Those circular DNA can either be chromosomes, and the chromosomes are really big, right? They consist of thousands and thousands and thousands of pairs of bases, or it could also be another region, another type of DNA called a plasmid. And plasmids are much, 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 much smaller than the chromosomes. They usually only have 1 or 2 or 3 genes on them. So, and that's very interesting. It's actually really interesting, plasmids, because bacteria don't really need to have these plasmids in them, but they can absorb them. So that's one of the things that make them interesting. If a bacteria didn't have its chromosome, it couldn't live. But it can live with or without these plasmids. And again, they're just much, much smaller. So, and they also replicate separately from the chromosome. So that's something else to kind of keep in mind. If we want to clone or copy a small segment of DNA, let's say we want to get this tiny little bit of DNA from this donor, foreign DNA, we will chop it out and insert it into a plasmid. We don't do it into the bacterial chromosome, too complicated. We do it into the plasmid. And then we take this plasmid, and because bacteria will just kind of randomly suck up these plasmids from the environment, we can make a new bacterium that has a gene in it that it never had in it before. And so we call that bacterium a recombinant bacterium. A recombinant bacterium contains a recombinant plasmid, and a recombinant plasmid contains recombinant DNA. Okay. So hopefully that all makes sense to you. And what we're going to talk about now is the steps in cloning a bacteria. So we've got this bacteria, this recombinant bacteria, right? And what you do is you kind of grow this bacteria in a culture, and it's going to go through this process of making the gene product. Now, the gene product tends to be a protein. And so, we use this word expressed. This is actually a term. The protein is expressed by the DNA, by the plasmid right here. And specifically, the one that we're interested in is this gene of interest. So what we can do with this bacteria, this recombinant bacteria that's making all these copies of itself, and in the process of making copies of itself, it could also be doing its thing by making lots of proteins. One of the things that we can do is we can harvest those proteins directly, and we can use them to help with human health. So insulin, for example, is an example of a protein that is harvested from bacteria. We used to harvest insulin from horses, but now we harvest human insulin from bacteria. So if you know anybody who is insulin dependent, you can thank bacteria for that, for helping them stay alive. Well, there's lots of genes that are done the same way. Human growth hormone is the example that this cartoon is showing you about. So if an individual has a growth problem, you can give them human growth hormone that was grown in a bacterium. On the other hand, there are other things that you can do with this cloned DNA. You can take this gene of interest, and you can get the many, many, many copies of the gene because we've allowed this bacterium to make copies of itself. And it can be used to insert genes into corn, for example, in this cartoon. And the gene that we're talking about here is the gene that allows corn to be resistant to being eaten by pests. And that could be a very good thing if you've got a lot of these pests eating the corn. You can kind of kill the pest without having to spray pesticide. That's got a lot of potential right there. So we could use genes to alter the bacteria themselves, grow up a lot of those bacteria, and use those bacteria to, for example, eat petroleum products that have spilled into our oceans. We could, on the other hand, we could use those proteins harvested to dissolve blood clots and allow people to live from having had a heart attack. Any time that you make multiple copies of a single gene, what you're doing is something called gene cloning. And just to reiterate, if you put those genes into a plasmid, that plasmid is called a cloning vector. And so gene cloning is used to make lots of copies of either a gene product, like a protein, or it's useful for amplifying many copies of the gene itself that can be used to apply to various different situations, like inserting the gene for pesticide resistance or making bacteria that are very good at breaking down toxic waste. How do we insert genes into, like this, a gene of interest into a bacterial vector, for example, like a plasmid? Well, we use something called restriction enzymes. Restriction enzymes are also known as restriction endonucleases, and I think that I have that on one of my later slides. But what a restriction enzyme does is it is an enzyme that can cut DNA, usually in a zigzag, but not always. We can see here it's cutting DNA in the zigzag pattern. And any place where a restriction enzyme recognizes DNA sequence, in this case the sequence is GAATTC, it will cut that DNA at those restriction sites. And it will make something called restriction fragments. So if you cut up DNA, then you've made fragments or pieces. And if it's cut by a restriction enzyme, it's called a restriction fragment. So if you have two different individuals, and this individual one has this restriction site in 3 places along the DNA, and this individual has the restriction site in 2 places along the DNA, you'll get a difference in how many different fragments you'll make and how many different size fragments you'll make. And that also is very useful when doing genetic engineering. So if you have a bunch of different fragments cut up from DNA and you put them into a gel, you can do something called gel electrophoresis. And we're going to be doing that in lab. So this is a gel. And you put your samples of all those mixed different fragment lengths into these region called the wells. And then as you run electricity down through this gel, what happens is that the DNA moves down from the negative cathode to the positive anode. It's moving towards the positive end. And that's because DNA has an overall negative charge. And so it's attracted to the positive anode. So it moves along. But the DNA doesn't move along at the same pace. It moves at different rates. The larger the fragment, the slower it's going to move through the gel, because this gel is kind of thick. It is porous, but it's kind of thick. And so the larger fragments have a harder time moving through. The shorter the fragment is, the easier it is to move through and the farther it will travel. So of these 4 samples, 1, 2, 3, and 4, these 2 individuals have 3 fragments and these 2 individuals just have 1. Now you might be asking, what's the stuff here on the side? And that's called a ladder. And basically what it does is it helps you figure out exactly how large your fragments are. So we'll be dealing with that in lab as well. Again, the whole point of this gel is to separate mixtures of different fragment sizes based on their length. That's what the gel does. Restriction enzymes are also useful besides just cutting DNA. It's actually useful for joining DNA from two different sources because restriction enzymes often have these zigzag cuts. And I showed you those zigzag cuts before. They have these zigzag cuts, and the zigzag cuts can lead to these unpaired bases being available. And those unpaired bases are called sticky ends. So if you have a sticky end and you take your DNA and you put it in the middle of a plasmid, it'll just line up really nicely with the plasmid, right? It'll have the T's match up with the A's and so forth. And then all you have to do is bring in a little bit of DNA ligase. Remember that from before? DNA ligase is what joins the ladder or the phosphate sugar backbone of the DNA together. So this end will get joined to this end in part. We've got that the A's match with T's and hydrogen bonds there, but we need this end to be attached to this end of the backbone, and that's the job of DNA ligase. So putting things all together, if I've got a plasmid, and in the middle of this plasmid, I've got a restriction site. The restriction enzyme will cut that restriction site and reveal these sticky ends. When the sticky ends are revealed, if you have a fragment from a different source, right? That's your, maybe that's the gene you're trying to replicate or to clone. You take this and you have to cut it with the same exact restriction enzyme so you can have the same exact sticky ends. Remember, there are thousands of different restriction enzymes out there, and they'll just kind of naturally be attached to each other because of our complementary base pairs. And then DNA ligase will seal those ends, and you have your recombinant plasmid. It's pretty straightforward. Now, it won't always work this way. Sometimes, like, these two things will seal back together, or sometimes, our fragments will kind of be attracted to each other. The ideal version of it is to get this recombinant plasmid, and that does happen quite a bit. After you do DNA cloning, you could retrieve that piece of DNA and make lots of copies of it in a machine in a process called a polymerase chain reaction, or PCR. So I'm going to describe to you now the process of polymerase chain reaction, or PCR. PCR can make lots and lots of copies of a target segment of DNA in a very short period of time. So if you have cut out your target DNA from your source using your restriction enzymes or restriction endonucleases, and you put them in this PCR machine, there are 3 basic steps that we've got to go through in order to make lots and lots of copies. And the 3 basic steps just take a couple of minutes total. So the first step, before I go into the various steps, I do want to tell you that one of the things that we're going to be doing is heating up our DNA. And so we need an enzyme that can withstand the high temperatures. Because, remember from before, enzymes under high temperatures, they don't tend to do very well. So scientists have been able to find a very, very heat-stable DNA polymerase called Taq polymerase. Now, this word is pronounced Taq, T-A-Q, Taq. And that's short for the name of the bacterium in which it was found, which was Thermus aquaticus. It's found in hot springs. So this is a bacteria that lives in hot water. It can handle hot temperatures. All right, so as I was saying, there are these 3 stages, and the first stage is denaturation. We've heard that word before, denaturation. In this case, we're using high temperatures to break the hydrogen bonds between 2 strands of DNA. And so when you break those 2 strands, what you have is those single-stranded DNA. the next thing that happens is that we're going to insert some primers. Remember, DNA polymerase needs primers in order to work. These primers are usually made up ahead of time. And so we just have a whole bunch of primers and we throw them in there with the DNA. And when the DNA denatures, the primers then anneal or join to the DNA. And once they are annealed or joined to the DNA, then the Taq polymerase, again, it's that special kind of DNA polymerase, will allow the DNA to make daughter strands. And the daughter strands are just copied. I'm not exactly sure why this green strand isn't drawn a little bit longer here. It's not like the DNA polymerase can make copies of things that it's not using a template from, but no, that's the picture that I found. I think the one from your book might be a little bit better, but I like the graphics on this one. So when the Taq polymerase or DNA, that special DNA polymerase is adding on the nucleotides, that's called extension. Okay, so denaturation, annealing, and extension. Let's take a look at how your book shows that. So with denaturation, very briefly heating those strands to separate them. Annealing is adding on the primers, right? And then in comes the DNA polymerase, and it builds in that 5' to 3' direction. Builds, it's going to build in the other side, the other direction, builds your new daughter strands. That's pretty straightforward and simple, and it happens very, very quickly. Over time, you went from having 1 strand to 2 strands, you can go from 2 to 4, you go from 4 to 8. Very, very short period of time, you can have millions and millions of copies of your DNA, and that's amazing. That's really, really useful. PCR does this in a machine. It doesn't rely on bacteria to do it. But then of course, after you do your PCR amplification, you still might need to make a gene product. You would still have to put that fragment back into a cloning vector in order to, you'd have to mix them together, you'd have to use DNA ligase to join them, right? But if you need lots and lots and lots of copies, then the PCR process is the way to go. Remember I talked about those primers earlier, and those primers are made specifically to find a site that matches the site for the cloning vector. So that's something that's useful. And I just wanted also to show you a picture of what a PCR machine looks like. Again, just a very simple machine. We have them at our school. And these machines have had a huge impact on biological research and genetic engineering. In fact, we can use all of these techniques to then do things like DNA sequence. So once a gene is cloned, you can sequence the entire genome of an organism, and you can find out all the A's, T's, C's, and G's that belong there. We can do this for organisms that are alive today, and we can do them for organisms that have been lost for thousands of years, like this woolly mammoth. Let's take a look at what DNA sequencing is. So DNA sequencing basically is a technique by which you determine the specific order of A's, T's, C's, and G's, are nitrogenous bases in a piece or a whole genome, a piece of DNA. When DNA sequencing first started out, it took a very long time to do. It was an extremely laborious practice. But in the last 15 years, we've come up with some very rapid and inexpensive techniques to sequence DNA. It's known as next generation sequencing techniques. They sequence by synthesizing complementary strands of a single immobilized template strand. So you take this template, you hold it in place, and you put letters in a row, and the sequence that is put in is being able to be detected by either chemistry or electronic monitors that identify which nucleotide is being added at which step. So what you see right here is a bunch of computers that are used to sequence DNA by sequencing, we say sequencing synthesis. So I just thought I'd show you what these different lines mean, and notice that there's some color coding. So they're kind of a yellowish color for C and orangey color for G. Sorry for those of you who are colorblind, a light green for T and a dark green for A. So if we look at this a little bit right here, we see it's a light green and then a kind of a yellowish color, and then a light green, and then kind of an orange color. But some of these lines are taller than others, and the taller the line is, the more of that base that exists. So if it's short here, there's only one base put in, and if it's about double that height, it's 2, and you could have 3 or 4 in a row. So this sequence right here can be read as T, T, C, T, G, C, G, A, A. And it seems very easy. I mean, coming up with the technique was very difficult, but in reality, anybody can run one of these next generation sequencing machines if you have a little bit of technique under your belt. We actually have started using a third generation method now, and the third generation method is actually even cheaper and faster than the next generation methods. What this involves is a little nanopore, which is a tiny little hole through which a single strand of DNA moves through. And as the single strand of DNA, it's kind of unwinding as it reaches the nanopore. As it unwinds, it moves through, and different nucleotides are identified depending on the distinct way that each nucleotide interrupts the electrical current across the pore. So a T will disrupt it differently from an A or a C or a G. And that's how you do the third generation sequencing technique. The last thing that we're going to talk about is how we can edit genes and genomes. And over the last 10 years, there's been a brand new technique that has come to light called CRISPR Cas9 system. It's very powerful, and it does things that are just very astonishing. And I'll talk to you about the technique, and then I'll talk to you about some of the applications of it. Cas9 is a nuclease, and nuclease is just something that cuts double-stranded DNA as it's directed to by something called a guide RNA. So the guide RNA is like a probe. It is complementary to the target gene. Okay, so here's your guide RNA, and it usually has a little loop there just to make it more stable. These two sides of the loop are complementary to each other. And then this right here is your sequence of RNA that is complementary to the target sequence of the DNA that we're looking at. This is your Cas9 nuclease. And in the Cas9 nuclease, you have these active sites that act as scissors and will cut both strands of the DNA at the same time. So you kind of put together the guide RNA and the Cas9, and then you expose this, which is the CRISPR Cas9 system, you expose it to your target DNA. And when the target DNA gets into the Cas9 system, it will like literally cut that DNA in half, or wherever the target DNA is recognized by that guide RNA. Researchers have used this for a lot of different things, including the ability to knock out, and that's actually a good term, knock out. You see that a lot when you get into genetics. All that knockout means is to disable. So it knocks out or disables a given gene. And what that's allowed us to do is to figure out the function of a gene. Believe it or not, we don't always know what every gene does, and we are in the process of learning what they do. This is true for humans, but it's also true for all the organisms that we interact with. Researchers have also been able to modify the CRISPR Cas9 system in order to repair a gene that has a mutation. This is both very powerful and very scary. So we'll talk about how it does it first, and then talk about some of the implications. So remember, we've got our guide RNA, and we've got our Cas9 nuclease, and there's our that part of the guide RNA that's complementary to the target gene. And then of course we've got our cutting, our little scissors area that cuts the DNA. So here's, if your target DNA has a mutation, then what happens is it cuts the DNA, and then we can insert some new DNA in, especially if we've got some sticky ends right there, right? Add a little DNA ligase, and you've just cut out a bad bit of DNA and replaced it with a good bit of DNA. And that's one thing that we've been contemplating, and actually have attempted to correct the genetic defect that causes sickle cell disease. Thousands and thousands and thousands of people die every year from sickle cell disease. If we could save their lives using this technique, that would be amazing to contemplate. Of course, that's not the only thing that people have done. And again, I'll talk to you about the bad stuff in a second. Here's another really interesting thing that's been done with the CRISPR Cas9 system. And that is that they have been using this insertion of genes to deal with insect-borne diseases. I mean, you've probably heard of a lot of diseases, including malaria, that are caused by insects. So what scientists have done is they have altered the genome of these insects, and they to prevent them from transmitting the disease. So what they've done is they've done something called adding a gene drive. So the blue insects are ones that have had their genome altered so that they cannot transmit. They've also been inserted with a gene drive. So if our blue insect mates with the black insect, then what the gene drive does is all the offspring of that mating result in them having both the inability to transmit the disease and the gene drive. That's not normally how things happen. Like, normally how things happen is when you have offspring, maybe half of them will have the trait that you want to pass on, and the other half won't. And so, but with the gene drive, it actually ensures that all the offspring get the gene that you want. And so the gene quickly establishes in the entire population. How does this work? Our blue insect here has the gene that prevents transmission of this disease and has the gene drive. And when a mating happens with an insect that doesn't have that insertion, what happens is that this gene drive makes a cut in the DNA, the homologous chromosome, opens that up and causes it to reproduce and have another copy of that gene inserted into that other chromosome. So it opens it up and inserts the gene. So you have two copies of it. And if you have two copies of it, you're bound to pass that on to the next generation. Here's a slightly different version of looking at that. So, and showing you the Cas9 system. So here's your Cas9, and here's the gene, your cargo gene, and here's your cut site. In normal inheritance, in this case, we've got a red insect that's got the Cas9 system with the gene drive and the blue insect that doesn't. The altered gene doesn't really spread. It's getting passed on to half of the offspring, but that's not spreading throughout the population. With the gene drive inheritance, every offspring has it. And so this one, between these two, all of their offspring have it. And pretty soon, after a few generations, the altered gene is going to be in the population. Insects reproduce really quickly. They might be reproducing every 3 or 4 weeks. In the case of mosquitoes, that does happen. So you could quickly eliminate diseases like malaria by using this CRISPR Cas9 system that along with a gene drive. Now, the last slide I have for you today is talking to you a little bit about the ethical issues surrounding the CRISPR Cas9 system. This person you see here is Jennifer Doudna, and she won the Nobel Prize in 2020 for her discoveries of the CRISPR Cas9 system. And basically, a lot of people are really excited about this because it can cure a lot of genetic disorders. It can cure a lot of insect-borne disorders. It has just so much potential. But, of course, it also has a lot of room for abuse. And one thing that has happened is apparently a researcher in a foreign country took the system and inserted the genes into babies that just inserted a gene to make them less able to get the HIV virus, which is, of course, the virus that leads to AIDS. Now, it probably did this with the best of intentions, but he also did it without the permission of those babies. And we don't know the repercussions of inserting these genes into babies because we just don't know enough about how this whole system works. So there is a lot of potential to misabuse things. For example, humans, we tend to certain cultures have certain ideals about what beauty is. Maybe, we in our culture think that being tall is beautiful. And so what if you started inserting a gene for height, increased height into humans? How ethical is that? I don't know. This is something that we definitely have to be talking about as a culture and as a group of scientists. And it's not something that we think that should be done lightly or taken lightly. So, but it is powerful, and it has a good chance of healing people and keeping people alive, but definitely has some major problems associated with it. I hope this has gotten you thinking a little bit anyway, and have introduced you to some important concepts of DNA technology. All right. Well, that's the end of chapter 13. I hope you enjoyed it, and we'll see you in chapter 14.